This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1047, recorded on September 22nd, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. Well, let's see. I'm looking out the window. It's partly cloudy. The temperature's in the middle 70s. Humidity is about 40%, somewhere in that neighborhood. An ideal day, basically. But trouble on the way. It's going to rain all weekend.
I'm looking at the weather here. Manhattan is 72 and cloudy, and there's a big hurricane moving up the coast.
Exactly.
My gosh, it looks like it's at Maryland almost.
Yeah, it's moving. It's whipping through.
So we're going to have heavy rain all weekend?
Afraid so.
I'm always inside anyway. What's the difference? Also joining me today from Austin, Texas, Rich Condit.
Hi, everybody. Well, we got 96 degrees, headed for 98. It's going to be 100 both days this weekend. And then maybe it'll cool off a little bit. There's a, in quotes, cold front coming through that will, on Monday, lower the temperature to 93 degrees.
Burr.
And the long-range forecast doesn't look like there's much of a break from this. We're not getting down to the 80s anytime soon. So there you go.
I decided
Texas.
I like hot weather, and I'm sad that I don't live somewhere where it's hot all the time. But
well,
maybe I'll come visit you for 2 days and get my fill of hot. What do you think of that?
We can fill you up with hot weather real quick.
There's a news show on NPR called The Evolution of Earth or something like that. And this week's episode was Life at Extreme Heats. And so they went to the Sahara Desert and they got video of the life cycle of what they call silver ants. I've never heard of silver ants before. They are the fastest moving insect in the world. They come out in the middle of the day when there are no predators around, like lizards and things like that. And they have special hairs all over their body that reflect the sunlight. So their body temperature is about 10 or 20% lower than the outdoor temperature really is. And they can't stay out for very long. Well, they captured a dead scorpion that died from heat exhaustion, and they had dismembered it in segments. A troop came in, took a leg, went back in the hole. Another troop came out, took another leg, went back in the hole. It was quite amazing to see. And the life form is actually one of the very few that's found in the middle of the Sahara. And then they went to Australia, and they showed zebra finches in 45 degree centigrade temperatures. And they were the only animal in the entire area that could find water. And they did. And it had some amazing adaptations. The mother finch, I'm boring everybody with this.
I'm just waiting to introduce Brienne.
Right
that's
there.
all.
The boring, I'm sorry, Brienne is interested in this.
I didn't introduce Brienne yet. Hold on. Joining us from Madison, New Jersey, Brienne Barker.
Hi, I do not have a lot of adaptations to the heat, but it's 72 and cloudy. Yeah, 72 and cloudy here, and it is going to be absolutely pouring all weekend, and I will be indoors and cozy with hot chocolate.
Right.
I just realized there's an app on my laptop called Weather. I didn't know that. I know there's one on my phone. So I have it open here. I have Manhattan, I have Scott, where I live, Washington, Madison, Hamilton, Chicago, Palo Alto, Zurich, Boston. I have all these cities on here. I have Austin, Galveston. I've been Munich, all the places I've been to. I have Tegernsee, Rich.
That's cool.
It's 52 degrees in Tegernsee, and it also tells you that it's dark. Hey, it's raining in Tegernsee. I have Montreux. This is so cool. It's like, yeah, I'm just discovering the weather app. Duh, idiot.
If you look in your phone and you swipe on the weather app, you should have those in your phone too.
I used to have the weather app on my front, and it disappeared. And now
So I have to tell you this. And let me just tell you the end of the story. It's very quick.
Okay.
A woman, she puts up with all those hot temperatures for the zebra finches, and she discovered how they cope with the heat. The mother finch on the nest does a high-pitched chirp over the eggs. She says, "What the heck is that bird doing a high-pitched chirp? What does that do?" Well, it's interesting because when the eggs hatch, the finch babies are on average smaller than finches that didn't chirp. Now, how did she determine that? Well, she collected a bunch of eggs when they were first laid, put them inside incubators inside of a lab at the same temperatures, and then played the chirping noise over only half of them. And lo and behold, and she mixed all the eggs up so that they all came from different females. And the ones with the high-pitched chirps were lower body weight than the ones that didn't hear the chirps, so they could tolerate heat better because they needed less water. Now that is incredible.
So throughout your whole big rap here, ants and finches included.
Yeah.
It's a wrap. It's a wrap.
I wind up. I'm thinking to myself, what is sentience? Okay, we've been having this conversation occasionally in the past. Okay,
yeah, we do.
You know, how do you? Is there a distinction between instinct and sentience?
Oh, that's a good
where
way
do
to
you
put
draw
it.
the line?
It's a very good way to put it. Yeah.
It's not for me to decide.
No, I don't think.
Just a messenger.
Probably nobody knows.
Well, right. Rich, there are people who have thought about it a lot more than we have. Okay, so that's the people you want to hear. So all of us have thought about viruses and parasites and immune systems, so we can talk, but we have not thought about the sentience. So for us to talk about it is not really useful for other people to hear because we don't know anything, right?
Well, you've got an evolution show on your thing. I would love to hear about rapid evolution.
Well,
this was really rapid.
I don't know about sentience. Anyway, let's get down to business here.
Okay. No.
Wait, we're here to do a podcast?
That's right.
We could just talk for an hour and probably some people would like it, but we have an agenda. And first of all, if you enjoy this, and I don't mean 20 minutes of banter. I'm sorry if you hate this. Just fast forward. We like to talk. If you enjoy this, our science programs where we try and put good science information out there, please support us. You don't have to give a lot. We have lots of listeners and viewers, and if you all gave a little, you could fulfill my dream, which is to make a company that endures, right? A science communication company. I was talking to one of my listeners, our listeners. She visited the incubator yesterday, and she says, "You need an organization chart. I'm going to make you one." So you need you, and then you need a COO who's going to oversee everything and make sure everything's on schedule. And then under that, all the podcasts with each of the hosts.
Oh, brother.
It's okay. It, you can do those things in PowerPoint very easily. So she's a corporate person. So she said to me, I actually don't think it's a bad idea to find a COO that can make sure everything is scheduled and paid for and blah, blah, that would be useful. I don't have money for a full-time person yet, but anyway, what was I doing? So if you'd like to support us, I have a vision to make this a proper company that exists beyond Vincent. Please support us, microbe.tv/contribute. And I was mentioning on TWiV yesterday, I had canceled my New York Times subscription last year because I was unhappy with some of their coverage. And I realized when people say to us, I'm canceling my subscription because of what you said, we get pissed off. So I thought, I did the same thing. So I resubscribed to the New York Times. There's actually a deal for $46 for a year. Although I promptly forgot my email that I used, so I have to go figure out how to get back to it. You'd think I was technology limited, So I'm not going to quit things when I disagree with them from now on. I have to put my money where my mouth is.
If you stopped your New York Times subscription, how do you play all the games on their website? I don't know what I would do without that.
Well, I don't play any of their games.
You should play their games.
But today my pick has to do with reading their articles for free. So, all right. So that's one thing. And the second is Amy Rosenfeld has a second technician position in her laboratory at the FDA. And I realized that for the last, I don't know how many weeks, the link has been broken. So it's no wonder she's not getting a lot of applicants. Oh my gosh. I feel so Luddite sometimes. And I know how to use this stuff. I don't get it. So this is a position for a technical assistant in the laboratory to do experiments. And Amy works on enteroviruses. She's very interested in pathogenesis, how they cause disease. She's interested in immune responses. So the assistant will help do experiments in those areas. And there'll be all BSL-2 conditions, so you don't have to suit up in any fancy way. And so there will be a link that works. I have to thank Charles, because Charles every week sends me an email. Professor, the link is broken. It's broken again. It's still broken. I have a special email called PETA@microbe.tv. You know the word PETA? Does anyone, do you guys know who, Rich? Dixon, you know what a PETA is?
Yeah, it's a piece of hollow bread that you put stuff in.
Okay. Anyway, PETA@microbe.tv. And if you find things wrong, they go there. And Charles is the most frequent emailer there. I appreciate Charles.
Is that what you actually type in, or does it just go there?
Oh, PETA@microbe.tv is the email address.
Okay.
And I have a special email. It's a email box that receives it. And several people use it, and they tell me when links are broken. And I'm just amazed at how many mistakes I make.
Well, your error rate is probably pretty low, given the volume of potential mistakes that get through okay. I mean, you do a lot.
Yeah, I appreciate it. It's not an excuse. I should get it right. Some of the things I don't like, every week there's a link to letters read, right? And sometimes it's episode 1046 and I have the link for 1045 and I swear I changed it.
But Vincent, you know that if a polymerase has too high a fidelity and makes too few mistakes, then that's not good evolutionarily. Right. So I think your error rate is probably exactly as it's supposed to be.
Huh?
But it gets people annoyed, Brianne. Perfect. I don't want to make people annoyed. I want people to be content.
They don't get annoyed.
So they
don't get annoyed. That's their problem.
Anyway, there you go. Those are our announcements today, and we have a snippet in a paper for you. And the snippet is about post-acute sequelae of COVID, past or long COVID. And we don't really talk much about long COVID, but I think this is an interesting paper. I'm sure that this is an interesting paper to discuss. It's in Nature Medicine. Benjamin Bowe, Yan Ji, and Ziad Ali. And they are from, let's see, the VA in St. Louis.
Yeah, basically.
Yeah, the St. Louis VA.
There's somebody from actually
Wash U.
6 is Wash U, yeah. That's the senior author has numerous affiliations.
All right, so everyone knows by now that you can get SARS-CoV-2 infection, and at a certain frequency, you get what we call post-acute sequelae, which means, so the SARS-CoV-2 infection is an acute infection. You have a defined onset and resolution of the infection. That's an acute infection, rather than a persistent, which would last the lifetime of the host. And these are post-acute sequelae. It's stuff that happens after the infection's over. Like, there's a whole array of sequelae in all kinds of organ systems of ourselves. And one of the things one of the difficulties is that there's so many of them, right? Many different organ systems, and not many studies have looked beyond a year. So, how long is long COVID, right? Is it a year? Is it 2 years? Is it forever? Well, we can't do that yet because we're only 3 years out. So this paper looks 2 years and does some very interesting conclusions. So the paper is basically taking data from the U.S. Department of Veterans Affairs. They have a national healthcare database, Now, veteran VA people are typically men, older men, and you'll see the numbers in a moment here. So it's not perfect, but it's a good system because, for example, they built a database here of 138,818 U.S. veterans who survived the first 30 days of a SARS-CoV-2 infection, and they follow them up for 2 years. And then they have a control group of 6 million users with no evidence of SARS-CoV-2.
My guess is that most of those people are pre-COVID.
Think?
I mean, if you really want it, well, I was wondering, I did the math to figure out that only 2% of their total study population had COVID, and that doesn't sound right.
Yeah,
okay.
Probably
so then I started to think, well, where'd these controls come from? And how can you be sure they hadn't had COVID? A good way to do it is to draw from people who were around even before COVID.
So basically, they have health records of all these people. They're stored in computer databases, right?
This reminded me of a past paper that had looked at one of the herpes viruses and neurodegenerative disease using a similar veterans database that had some immense number of individuals. And I was so impressed with it then, and I'm remain impressed with the whole idea now.
Well, one of my reactions to this was about this database. When people talk about the notion of what do they call it, single payer, universal health care, one of the and whether or not the government in general or the U.S. government could pull this off, quite often the VA system comes up in the conversation. All right, and I've heard both positive and negative things, but a lot of positive stuff about how well the VA does. But no matter how well they do, having all of this stuff in a database is so valuable. And outside of the VA, I don't know that there's any, there's no nationally integrated.
No, the insurance companies have it.
Like Kaiser Permanente, right?
They've all got it.
So they've got a fraction. They've got a fraction, and they're keeping it to themselves. It would be nice to have this on a national level.
Right.
It would, because there are not a lot of women in this study, and they're not only the average age is like 60 something. So
you'd have to move to England to get something like that. I think, I'm afraid.
Well, that was another thing that we found out during the pandemic is that places that do have a national database, like Israel, and England, you can get a lot of really valuable data about what's going on.
Sure, I remember also that the Netherlands was prime for the data that suggested that vaccines don't cause autism, because they kept track of everything, and they know when they switched from thymol to no thymol, and there was no difference. And obviously, that's it's a little bit, I guess, in some ways invasive, because people look at it as an incursion into their privacy. But if you grow up in that society where everybody shares, like for instance Iceland or some other isolated countries like that, then those databases are quite useful.
We have done some papers where they use Kaiser Permanente databases. I think there was one where they looked at the shingles vaccine efficacy, right?
Yeah.
So sometimes they do share it, and that's good. But we should have a national database that would really be useful to look at. Well, anyway, they look at 80 pulmonary and extrapulmonary sequelae of SARS-CoV-2 for 2 years, right? And I had COVID in November-ish, and I don't recognize any of these in me, although maybe it's not for me to diagnose myself, right? But I think a lot of these are self-reported. I don't know that there's a diagnostic test for a lot of them, but I guess I'm lucky that I don't have any of them. Okay,
so this is the other thing that occurred to me is that in the database, there must be codes for all of these different conditions, okay? Because nobody's going to go through 6 million records by hand and figure this out. It must be that the system, and I don't know how the medical profession works and how widely distributed this is, but certainly in the VA system, when you report with this or that, it must get recorded with a code of some sort.
I think there are standardized codes that are used across the board.
I mean, I just went for an annual physical, and everybody I interacted with had a big computer screen in front of them, right?
That's
and
right.
then I go in the exam room with my doc, and he's got a big screen on the wall, so I can see everything he's typing in, right? On all
I thought he was watching the soccer game.
You know, it's funny because I said, "What do you do for earwax?" And he went and he searched for something on Google, and they blocked it. They wouldn't let him search.
You're kidding.
He said, "Oh, they're blocking me. I can't." But here's the name of the thing.
That's funny.
Isn't that funny? They
Yeah.
should have a database of stuff. You know, it's an over-the-counter thing.
Right.
Anyway, here are the numbers. So, as I said, 138,000 SARS-CoV-2 infected people, 6 million controls. The COVID group had a mean of 60.9 years, and 11% were female. And the control had a mean of 62.8 years, and 9.9% were female. So there you go. Right away is a weakening or a shortcoming of this study, right?
The control groups and the test groups are comparable profiles, but they're not sort of a cross-section of the U.S. population.
No, this is the VA, right? Veterans. So they looked at risks of death, hospitalization, and 80 pre-specified post-acute sequelae on their own, and also by organ system. And then they separated them by hospitalized and non-hospitalized people to see if they found at different time points. And this could really be a summary, but I do want to just go through a little bit of this because the bottom line, you'll see, is quite clear. Basically, if you get hospitalized, you're going to have long COVID for a long time, at least because it doesn't get better in these 2 years. Most of it doesn't get better. Okay. Compared to the control, people with COVID who are not hospitalized were still at an increased risk of death from 91 to 180 days, but not 180 to 720 days. So they call this the risk horizon, which to me is a little poetic, right? Remember the event horizon?
Mm-hmm.
It's kind of like the risk horizon. So you will not die if you make it past... what is it? 181.
So life is a black hole, is what you're trying to say?
It's a good way to put it, Dixon. I like that.
Because you disappear, but you never come back.
All right. So in the non-hospitalized group, over this 2-year period, the risks of getting all these other conditions, I'll go through just a few of them in a minute, became not significant for 69% of the examined sequelae. So all these organ things that they're looking at, sequelae, not things, sequelae. In the 2 years, the risks declined to a point where they're no longer significant for just 69% of the sequelae, but they remain high. The risk remain high for 31% of the sequelae, including cardiovascular, coagulation in blood, endocrine, gastrointestinal, general, kidney, mental health, musculoskeletal, neurologic, and pulmonary. What else is left? Right.
Hangnail.
Oh my gosh. So the figures are very cool, right? And they're all coded. So they have abbreviations for all these things, which I'm not going to tell you, but here's the ones that are not coded. Death. Okay. That's pretty clear. Hospitalization, acute cardiac distress, or is that what's ACD? And that's something with the heart. Angina, AFib, aflutter, bradycardia. So these are all cardiac things. So there's like 30 different cardiac things. Then we have abdominal pain, diabetes, pancreatitis,
cardiac arrest,
constipation, myalgia, joint pain, depressive disorders, right? Liver abnormalities, Alzheimer's. My gosh. Osteoarthritis, myopathy, ischemic stroke, hearing loss. My hearing loss started before I had COVID. You know, there's a name for that.
I'm sure there is.
Age-related hearing loss. Because I saw it on the screen at the doctor's office. Prebiscosis. What is it? Something like. Hold on, I got to find this now.
I've got it. I know I do.
Presbyopia. No, that's
no. That's eyesight, right?
That's eyesight. Age-related hearing loss.
All the physicians listening right now are laughing their heads off.
I know they're laughing, but I'm a virologist, okay, and I admit it. Presbycusis, age-related hearing loss.
Presbycusis?
Yeah, PRESBY CUSIS. Age-related hearing
Right.
loss. So anyway, there are a lot of vision abnormalities. There are a lot of conditions that they're tracking here. And it's actually remarkable that, I mean, not one person doesn't have all of them, right? But you have a subset.
In the veterans, though, did they break out which ones that actually had active service in places like Afghanistan?
No, they're one of the in their little section about weaknesses of the paper. One thing that they point out is that they didn't try and dissect the populations by any particular criterion beyond just hospitalized or not.
I think
or where they caught it.
Right.
No, well, another thing is vaccination status, right?
Right.
Yeah.
And vaccination status.
Yeah, that was one thing that I was thinking about when looking at this paper. It looks like they basically got a group of about 6 million individuals and then followed them. And they picked this group in early 2020. So they couldn't have really started to think about vaccination. They started this pre-vaccine. So I would hope that maybe in a future year or two, a similar study looking at how vaccine impacted this could happen.
Right.
Being a veteran, isn't it required that you had vaccines?
If you're no longer in the service, if you're 60 years old during a pandemic and it's SARS-CoV-2, I can't imagine that there's any sort of vaccine mandate. If you're in boot camp or something like that, sure, they're going to shoot you up with all kinds of stuff.
So the age relatedness is also a problem here, then.
Yeah.
Mm-hmm. Yeah.
A big problem.
Yeah, it could be if you looked at 30 and 40-year-olds, you get a very different picture. Right?
Exactly.
I'm sure you would. So this gives a biased view, right? Because in the end, you're horrified at the extent of how long a lot of problems continue at a high percentage, yet it's 60 years old, average age, right? So if it's an average of 60, there are a lot older people and there are also a lot younger people, right?
Well, I don't want people who are 60 to have all of these problems anyway.
No, thank you.
We don't. I don't want anybody to have any problems. But, you know, humanity is apparently frail, right? Oh, my gosh. I mean, I was in this morning to get a blood work, and I'm just looking at everyone coming in for all this stuff. And I'm like, oh, my gosh. Is it our fault or is it just our bodies break at a certain age? You know, it's just, and then you look at all the machines. Oh my gosh. I'm looking at these big MRIs and, oh my
That's
gosh.
right.
It's just names of stuff, bone scans and thyroid scans and this scan. And that's, holy crap. It's such a, you know, I don't go much for this stuff. You can see that when I do go, I freak out. Oh my God.
One of the football games per year, it's a professional football game.
Mm-hmm.
And all of the professional football games at that day, everybody holds up a sign if you knew someone or if you yourself had cancer. And I guess about 80% of the audience held up cards.
Sure.
Yeah. That's 60,000 people on average per stadium. And you look at how many people have actually.
Yeah, it's very common.
It's incredible. That's not frailty necessarily.
Well, they say 1 in 3 people. If you're at a party, 1 in 3 people probably not only have your own birthday. I think 1 in 10 is your birthday, but 1 in 3 have cancer. Yeah. It's amazing. A consequence of living past 30.
Indeed.
All right. So so far, we're still looking at non-hospitalized people, right? So basically, some things get better, but not everything. All right. And then the other thing they look at, which is very interesting, is they look at disability-adjusted life years, daily, DALY. Okay, this is a very interesting stat, and I looked it up, and there's a nice WHO site. So DALY basically represents the loss of the equivalent of 1 year of full health. All right? So one daily, you lost 1 year of full health. And so then they said, how many dailies are we losing in these 2 years that we're looking at these people?
Oh, brother.
So there are 70, what is it? Here, there's so many pasks per 1,000 people, corresponding to 80 dailies per 1,000 people at 2 years. And we're looking at the, 180, 140,000 people. So 80 per 1,000, 80 lost years of work per thousand because of, well, associated with this condition, right? You don't know if other things are fitting into that too. And they break it down by neurologic, cardiovascular, et cetera. They look at first and second year DALYs, and 23%, 25% DALYs are due to the second year, and the first year is 75%. So that first year for the non-hospitalized is really bad. All right. So I find that a very interesting statistic that kind of makes it more real, right? It's a whole year of lost what again? It's a whole year of lost health, full health.
Yep.
All right. So how about the hospitalized group? So if you're hospitalized during acute COVID, you remain at risk of death and hospitalization throughout 2 years. And I think I mentioned this last year. There's a huge, for hospitalized people, especially those who go in ICUs, huge risk of dying even after you're discharged. Daniel said it last week, we clap out the patients who are discharged from the ICU, but we shouldn't be, because 30, 40% are going to die within the next year.
Is that number very different for COVID patients who come out of the ICU versus non-COVID patients?
I don't know.
Or is that just ICU?
Good question.
That's a good question. I didn't ask him that. But yeah. So over 2 years, the decline in risk difference between the COVID group and the control was much less pronounced in people who are hospitalized. So remember, the non-hospitalized people, there's a big drop in some of these symptoms, but not so much for hospitalized people. But some risks did decline. Some of them, the risks declined at 2 years for 35% of the sequelae, not the people, but the sequelae, and remained increased for 65% of the sequelae. And among those hospitalized, the risk remained increased for PASC in all organ systems that they looked at. And the DALYs, let's see how the DALYs look. 642 DALYs per 1,000 people at 2 years. Wow. 642, that's way more than we said for the other.
Much more.
Right? It was less than 100 for the other.
I mean, that's more than a year per person.
Yeah.
Exactly. How many were smokers?
They don't say that here. It's a good question.
I'm looking at this. What is it? Figure 1, I think. Mm-hmm. That's sort of the primary data that has all the colored boxes.
Right.
And I'm noticing that the incidence of bad news in the hospitalized, the incidence of disorders of one sort or another in the hospitalized group during the first 90 days is much higher than the non-hospitalized group. And then I go to myself, well, duh, these are, among other things, probably underlying conditions. Okay, because so for the one that caught my attention first was alcohol use disorder. Okay, right. COVID didn't cause alcohol use disorder. No. Alcohol use disorder was probably one of the factors that landed them in the hospital when they got COVID.
Yep.
Yeah. And, this whole idea of landing in the hospital, Daniel was always saying, Paxlovid reduces the risk of getting in a hospital by 80-some%, right? So you could make an impact on long COVID by taking Paxlovid, and we know that a lot of people don't get it for various reasons, and that are not good reasons. So that's something to consider. So that's an idea of the picture here. So here's the summary. People who are not hospitalized during the acute phase, and that's most of the people, by the way, that have COVID in this database, the risk of death becomes non-statistically significant at 6 months, and the risk of hospitalization remains elevated until 19 months. So even if you're not hospitalized, you could be hospitalized until 19 months when the risk for that goes down. Those people hospitalized, the risks of both deaths and hospitalization remain statistically significantly elevated throughout the 2 years. And at 2 years, risks remain elevated for 31% and 65% of the sequelae in non-hospitalized and hospitalized individuals. Okay, so that's the side-by-side comparison. And finally, the dailies, 2 years past, contributed 80 dailies per 1,000 people among non-hospitalized and 642 dailies per 1,000 among the hospitalized individuals. So that's a big impact. And then they go on to talk
Huge.
about, what this is a big problem for the healthcare system, right? Because
Enormous.
you have to take care of these people who have all these issues, and they call
That's
it a
right.
difficult and protracted road to recovery. And long, healthcare system is going to have to absorb this. So we don't have any way to treat any of this so far. So they say, well, we should prevent it, right? But
so I have a few sort of reductionist comments here. Well, one is, I mean, it may be obvious, but this comparing hospitalized and non-hospitalized is a way of saying how bad did you have it,
right. Right.
So, and in both cases, there are some things that linger. In general, with most of these things, over time, there is an improvement. That's the good news. The bad news is that some of these things can linger for 2 years. Okay. And if you had it bad enough to land you in the hospital, if you had a worse case, the chances that it's going to linger for longer are greater. So that's sort of my summary. Now, there was one thing that stood out to me, and that was loss of smell, because the data in those boxes looks different than the others. There's no difference, or there's not much difference between the hospitalized and the non-hospitalized, and it lingers longer. Okay, so that I thought was really interesting. My other comment is that nowhere in this paper do they use the term long COVID.
That's right.
Okay? But it's I think that's basically what we're talking about. And I think it's important that they don't use that word, because I think that these are the kind of data. I mean, nobody knows what long COVID is, right? Well, maybe that's an exaggeration, but there's debate about how are we going to define long COVID. And I think these are the kind of data that are going to give a definition to long COVID in the long run.
Yeah, this is not a single disease. This is post-acute sequelae, right?
Yeah.
A lot of different things that can continue after the acute phase. And long COVID implies, as you said, that it's a single disease, and it's not. And also it's not just COVID that does this. Other infections can give you post-acute sequelae as well. So it's just that we just went through this huge pandemic where a lot of people were infected. So it's in our face, right?
Yeah, we don't necessarily have a database of 6 million individuals to look at easily for some of those other infections.
That's right. We should, though.
There's one thing. There's another one here. I mean, you can pick individual things out, but this one, cholangitis, which is inflammation of the bile duct system,
right,
is really elevated in the hospitalized people and lingers to a very significant extent. And there's no significant difference between the control population and the people who weren't hospitalized. That's striking.
Yeah.
Another point they make, which is listen to this is really, really important. It's clear that the burden of health loss will not only impact patients and their quality of life, but also potentially contribute to a decline in life expectancy, because, beyond 2 years, and may impact labor participation, economic productivity, and societal well-being. So, yes, I agree. All those things can be impacted if you have these for many years, right?
Yeah, I was showing the life expectancy in the United States over the 20th century graph in one of my classes recently, and you can so easily see the 1918 pandemic there.
Yeah.
And I made the comment of, I wonder, in 100 years, what they will be saying about this pandemic, and showing similar graphs.
Oh, yeah, I show that one too. It's a big dip. And I asked the class, what did this? Most classes can get it, at least one person.
My first reaction to that is that we got so much other stuff going on that might impact on life expectancy that maybe SARS-CoV-2 will just fade into the background in 100 years.
Yeah, I mean, who knows?
There's a scary thought.
But given how frequently I show that graph, I do want to know about that same graph in 100 years.
Yep.
Well,
I'll report back to you.
Perfect. Thank you.
There are already some data, right, on reductions
Yeah.
in life expectancy. You could probably make a new graph, right?
Probably.
But the thing is, it's different for different groups of people, right? Depending on their exposure. Anyway, the other thing is I wanted to read what they say about their limitations. A lot of limitations, but the one that's, I think, important, the VA population comprises those that are mostly older and male, which may limit the generalizability of study findings to the non-veteran population.
Right.
We did not examine the effect within subgroups. And that last sentence, the results may help inform post-COVID care strategies and health system capacity planning to address the post-acute and long-term care needs of people with COVID-19. So, yeah, it is. It's a real thing, and it's diverse and different. And I don't think, it's another year is going to make much of a difference, right? Because if 2 years you see this, it's just going to slowly decline, but it's going to take many years.
Yeah, I also really like that they point out that they were specifically looking at 80 sequelae, which is amazing, but that theoretically there could be, as they say, yet to be characterized sequelae.
Sure.
That are not analyzed here.
Anyway, I feel lucky they don't have any of these, as far as I know, having had it once. But I know N of 1 is meaningless, but I can still feel lucky, right?
Sure.
and
Sure.
grateful.
You should every day.
I asked my doctor yesterday, are you going to retire soon? He said, no, it's a privilege to do this. I like that. I thought that was a good answer, right? Take care of people. All right, so that's like a public health paper. Right now we have a basic science paper.
Uh-huh.
So if you want to check out, don't, because you'll learn something here. If you're interested in influenza, in particular avian influenza viruses, listen, it's interesting. This is a Cell article.
This also has, in my mind at least, echoes of a lot of previous conversations that we've had about controversial experiments.
Yeah.
So hang in there.
Airborne transmission of human isolated avian H398 influenza virus between ferrets. And yesterday, Daniel complained about airborne because he said, some people use airborne for certain kinds of droplets. And here, obviously, they're just talking about through the air, which is what airborne means, right? Through the air.
That's right.
But some people use it to mean the tiniest of droplets that go far.
I think that is aerosol.
I know, but some people use airborne.
Yeah.
And there's a new nomenclature that's being proposed to get around that. And Daniel talked about
We spent many hours during the pandemic going over the semantics of this. And trying to bring some definition to it. And yeah.
All right.
I signed a reading for it for Wednesday.
Very good. All right. The authors are from, let's see, authors contributed equally. Number 6, we have Honglei Sun, Han Li, Qi Shi Tong, Shi Shi Han, Jiu Lu. Yeah, those are the co-first authors. And then the lead contact is George Gao, who is former head of China CDC, stepped down recently, very visible during the pandemic. So they're from the China Agricultural University in Beijing, the Chinese Academy of Sciences in Beijing, the CDC, China CDC, and University of Nottingham in the UK. Avian influenza viruses. I recorded an H5N1 episode a few weeks ago. It has to be released yet. But, you're hearing a lot about avian influenza viruses, right? Especially H5N1, high-path H5N1. But there are others like H7N9, H9N2. These are all subtypes. So the influenza viruses are categorized by the kind of proteins they have in the virus particle, what you would know as spikes. But there are 2 different kinds of spikes in influenza virus particles. One is an HA, one is an NA. So they get numbers like H5N1, et cetera. And historically, the epidemics and pandemics in people only involve H1, H2, and H3 hemagglutinins and N1 and N2 neuraminidase. But, these avian viruses have different ones. And so should we worry? And that's what this paper is about, is they're doing some experiments with avian influenza viruses from both birds and humans and asking, should we worry?
I want to make up the point up front that these influenza viruses, well, first of all, they have a segmented genome where each segment more or less corresponds with a gene so that they can do genetic rearrangements with each other with great facility. And the other thing is that we may talk about H5N whatever, okay, or H3N8. There's another 6 genes, okay?
Yep.
And those, I don't know, you may know, Vincent, to what extent those co-vary with the hemagglutinin and neuraminidase. But the point is the hemagglutinin and neuraminidase, as we will see, is not the only player in the story.
Yeah, they can independently be assorted, although there's sometimes some connection between the hemagglutinin and neuraminidase, because depending on what receptor is recognized by the hemagglutinin, the neuraminidase has to hit that receptor. So if the two are hitting different things, it's not going to work.
And I would think that, okay, so I'm making this up, but I would think that the hemagglutinin and neuraminidase is going to have an impact on host range. And
Yes.
So what the other segments that encode the polymerase and the matrix protein and all that kind of stuff may work better in different environments. Okay, so they may actually co-vary with the hemagglutinin and neuraminidase, because if you're going to infect this kind of cell, you're better off with this polymerase.
Yeah.
Okay.
Yeah. Well, we'll see an example of that here today. So what do we know about H3N8? So the first human so these are bird viruses, but April 2022, there was an infection of a 4-year-old boy in China, first human case of infection with H3N8. The boy got severe respiratory distress but recovered. May 2022, another H3N8, a different isolate was diagnosed in a 5-year-old boy. He developed mild symptoms that resolved. March 27, 2023, a third case in Guangdong. The patient was a 56-year-old female, and she died. All of them had exposure to live poultry, suggesting it to be the source. So these viruses circulate in poultry, right? And so that's the risk factor there. Now, we think that currently circulating H3N8 viruses are actually triple reassortant. So 3 different viruses are contributing to the segments of H3N8. And these include a Eurasian avian H3 gene, a North American avian N8 gene, and H9N2 internal genes, so non-HA, non-NA. So, co-infections of hosts by different influenza viruses give rise to unique viruses that have different property.
I also want to comment that you described the individual cases that have been described.
Yeah.
They don't state specifically, but implied in this is that there's no evidence at this time of any human-to-human transmission. This becomes, that's the question.
Yeah,
right.
Basically, but there's no evidence that has happened. I also wonder, this happens with influenza viruses all the time, is that they, different viruses are probing the potential, that's a little anthropomorphic, for infecting humans and even transmitting. People are getting infected with this stuff all the time, and they may shrug it off, they may get sick, whatever.
Yeah.
It would be interesting to know what the real incidence of infection with these is in human. I don't know. That'd be hard to determine. You'd have to look for, I guess
serology, right? You want to do
you do serology, but a lot of people are going to have H3, right?
Well, but they'll have human H3, but as we see in this paper, they don't cross-react, right? So that might be okay.
Okay. Yes. Okay. So you'd be able to tell.
But that's a good point that I think there might be more infections than we
Sure.
know of. These were just
Yeah.
symptomatic, right?
Yeah.
Yeah.
But maybe if you looked in people who work with poultry, you would find a higher rate of infection. In fact, they say in 2022, they frequently detected H3N8 viruses in chicken farms and live poultry markets in China. So, and this virus, H3N8, is well adapted to chickens.
Yeah, these are probably the only symptomatic and also diagnosed. You might imagine that there might be people who just
Sure.
have stuffy noses, but aren't getting diagnosis.
So this paper asks the question, should we worry about these viruses? Is there any evidence that they pose a threat to humans? That's a hard question to answer, right? Because we can do certain experiments, as you will see, but we still don't know if they're a threat or not. So let's go through this so you can get an idea of what kind of experiments people do to do risk assessment. And, we have to do these. Otherwise, we don't get any information. So don't say it's dangerous, please. You work on them under the right conditions, and you can do it. But if you don't do it at all, then you're in the dark, and you're going to be surprised. So first, they have some isolates from humans. So these H3N8 that have been isolated from people who have been infected, and they have isolates from chickens. All right. And they just infect cells of different sorts to say, do we see any differences between the chicken and the human isolates? And the human isolates are HN and CS, and the chicken isolates have CK in the beginning. So that's easy, CK. HN, I guess, human. I don't know, CS. Crime scene, okay? So the first thing they do is they take some different kinds of cells. They take organotypic differentiated primary normal human bronchial epithelial cells. So kind of organotypic cultures. They take cells from people and grow them in culture, and they look like differentiated cells. Then they have
these are what I relate to historically as raft cultures, right?
Raft cultures, yeah.
Where they take primary cells from human bronchi and basically culture them on a membrane.
Yeah.
And then ultimately create with that membrane a liquid air interface that mimics the conditions in your airway, and the cells, the primary cells differentiate into a ciliated epithelium. This boggles the mind. So the idea here is to do the best you can to approach in vitro in a culture something that looks like cells as they exist in a person.
Yeah, you put them on a membrane. Underneath is medium, and then the top has medium originally, and the cells get confluent, and then it's rich as you remove the medium, and the cells are like, oh, air. We can be ourselves now, and they differentiate. It's very cool. Then you can put virus on them and see if it's released underneath by sampling underneath. And then they use a cell line, a carcinoma of the lung cell line. They're called CALU cells. And they infect them with these different viruses. And the first thing they do is they use antibodies to look for viral proteins in the cells as indication of infection, right? They look at antibodies to the nucleocapsid protein. They look at markers for ciliated cells. They do fancy fluorescence microscopy. And they can look at the percentage of cells that are positive for infection. And basically, if you use the human isolates, HN and CS, you get a higher percentage of cells that are infected compared to the chicken isolates, right? So what would you call that? They're more infectious?
Who knows? Now you're going to get me into this. No, I'm not taking the bait.
Okay.
They are better adapted to infect in.
Because what you're taking the same amount of virus and putting them on each, right? And you get more infected cells with the human viruses than with the chicken. Yeah, they're better at infecting humans.
So what are they? I mean, okay, you asked the question. They're staining for viral NP.
Right.
And my guess is that is new virus NP. So in order to do that, they have to not only get in the cells but make NP.
Yeah, they're making.
And it doesn't
NP,
differentiate between whether or not they're getting in, or I mean, it could be they're getting in efficiently, but in some cases not replicating enough to make enough NP to give you a signal. So
would you call that infection if they get in and make some proteins?
You're the virologist. I'm retired. You tell me if they get in and don't.
I
make
think
NP.
infection means the virus gets in and starts to make.
Yeah,
some...
isn't that abortive infection versus productive infection?
Okay, yeah.
So
sure.
you could say a productive infection. Yeah. So this is a thing we're debating for the textbook now. What is infection? Right. I think it's different.
Oh dear.
I think it's different in cells and in hosts. Actually,
let's put it this way: it is a qualifiable word.
Yes.
Okay. You've offered several qualifications, and I think that those probably need to be used.
Yeah. So in a cell culture, you would say infection means you make infectious virus.
Yeah.
So that would be productive infection. So that would mean abortive infection.
So you want to qualify it. That's fine. That's totally fine. Productive infection. Right. So here we're not doing that yet because we're just looking at antigens that are made. But then they do look at production of infectious virus. They do tissue culture infectious doses. They basically ask, they quantify infectivity. It's not a plaque assay.
Plaque assay?
They don't do a plaque assay, but TCID 50s are okay for.
They're close enough.
They're close enough.
Okay.
And again, the chicken isolates, they make less virus than do the human isolates, right? Like the 100 to 1,000 times higher infectious output of the human viruses compared to chicken. So that's a productive infection, and the human viruses are better at it. And these are human cells, right? So the implication is that they're better adapted. It would be interesting to try some chicken cells, wouldn't it?
Yeah. What temperature? What temperature are you doing this at too? Because chicken's body temperature is higher than mammals.
At some point in this, they actually test a couple of different temperatures, but I forget.
These are done at 37.
That's not high enough.
Is it also in the GI tract in the chickens?
Yeah, it can be.
So would you need a GI cell line?
Good point.
You know, it really depends on the virus, though. Because I asked Ron Fouché that, and he said whether it's GI or not depends on the avian influenza virus. So I don't know about these H3N8s. That's a good question. But anyway, I'd like to have some chicken cells here to see if the chicken viruses are better at those, right? As opposed to just being generally poor.
Right.
If you know what I mean. So anyway, I'm not going to look it up. We'll have to. I didn't publish that episode with Ron yet. I have to do that. So the infectivity tells you that the human isolates do better in human cells. They infect better and they produce infectious virus better. Okay, how about ferrets? Now we're going to look at ferrets.
And remember, they're not people.
Ferrets are not people. I have a ferret behind me. Look, let me get it.
Do you really?
Yeah.
An actual live ferret?
No, it's not live. How would I have a live ferret here? Look.
Ah, that's so cute.
It's from a company that does contract research, CRO, a contract research organization. And I met them at ASV last summer, and they came to TWiV, and they gave me this.
Isn't that nice?
And they said, you have to show it on TWiV. And I did last year, but here it is again. Ferrets are not humans.
Nope.
Are you gonna skip over the mice, Vincent? Because they did mice too.
I'm sorry. Thank you for holding me back. Let's do mice first.
You're really galloping towards the ferrets, right?
Wait. I have a mouse here too. But look at this. This was made by, I think Martha made this. Look what she put on the back.
Mice lie, great.
That's also not a Balb sea mouse.
Because it's brown. It's gray, right?
Yeah. DBA2. DBA2.
It's deep. Isn't it C57 black? Anyway, mice lie. Monkeys exaggerate. I don't have a monkey, and ferrets are not people.
Ferrets they just sniffle.
Oh, I'm so happy to have. What is Brienne going to show us? You got something? You got a monkey.
Oh, you got a monkey. All right, there you go.
Well, you folks listening, I'm sorry you're not seeing the show and tell. We have all kinds of fluffy toys. I have a mouse and a ferret.
What the hell have I got?
And
I've got a giraffe, but he's back in my bedroom.
I got a baboon.
I have the same baboon somewhere. Yeah.
You
That's
do.
from
I gave
Ashley.
it to you.
See how much fun science is, everybody?
We're in a good mood today. Anyway, they first look at Balbc mice, which can be used to do influenza. They intranasally infect Balbc mice, and they first look at the H3N8, the human-derived H3N8. They get clinical symptoms: depression, decreased activities, and ruffled fur, body weight loss, and the lethal dose 50%, 10 to the 5 TCAD50. If they infect the mice with the chicken-derived H3N8, no clinical signs, no weight loss, no mortality. What's depression in a mouse?
I don't know how you define that.
Brianne, have you ever heard of that before?
They don't even want to go to the opera.
No.
Yeah. From things that some of my colleagues do, I think it's related to the level of activity.
Yeah, I would think so.
But I'm not sure.
They probably eat less also.
It's not that they're sad. They're just subdued, right?
Yeah.
Yeah, someone asked me that on the live stream the other night. What do you mean when you say you have to take them to the opera? And actually, I had to think about it because you got to see if they appreciate the opera, right?
If you did, you'd take them to Der Fledermaus.
That's good.
So
that's one of those sequelae that they did not look for in the other paper, was opera enjoyment.
The whole point is that there are potentially subtle effects of some of these things that you can't measure easily. And the joke is, have you taken them to the opera to see if they appreciate the opera?
Right.
So there, TWiV, what is this, 10:47? An hour or so into this? Yeah, an hour and 5 minutes. That's our place mark for taking mice to the opera definition.
Right.
All right. So then they want to see how the viruses reproduce in mice. Again, they infect them intranasally and they take out tissues: turbinates, lung, brain, spleen, and kidney. All the viruses replicated in the turbinates and the lungs. The human viruses replicate to higher titers in the turbinates and the lungs. Interestingly, the human viruses were also in the brains of the infected mice.
Yeah, that stood out to me.
In fact, Ron Fouché said that these recently circulating H5N1 viruses also end up in the brains of mammals that they infect. They're neurotropic. Interesting.
So this is a little difficult for me to interpret because mice are not ferrets and they're not humans and they're not chickens.
Yeah.
Okay?
Yeah.
But generally, these viruses, are by these criteria nastier in the mice than the chicken viruses or the standard human H3N2, which is another one of their controls.
Lung lesions are more severe in the mice infected with the human isolates. Histopathology scores more viral antigen positive cells in the human viruses. So I guess you could say, okay, in mice, the human and the chicken viruses have different phenotypes. So if we get isolates from people and we put them in mice, we might be able to say what's happening with them instead of using ferrets.
They did some sequencing work.
You could do that too. All right, so now we use ferrets. Mustela putorius furo. Wow, I didn't know that. So ferrets are susceptible and they display symptoms that mice do not. They sneeze and they cough, and they get fever. So ferrets are useful models.
They're kind of the aside from humans, the gold standard is a lab model for flu.
They're a good model, yeah.
I think they're pretty good for a lot of respiratory viruses. Isn't some of it about their ability to do respiratory transmission in lab studies as well?
Don't infect me.
This is Uncle Vincent talking.
I can have a sense of humor, all right? I can have a sense of humor. I like fuzzy things like this.
Very good.
So think about what it was like before we had video.
Exactly.
No, we couldn't do any of this stuff.
Or stuffed animals.
So the question is, Brianne, is an improvement? Is it good?
Right.
I think it's an improvement.
So you can knock ferrets intranasally. You take a pipetter and you drip it into the nose. You're going to have to anesthetize them because they're not going to sit still while you do that. They're going to want to bite you and scratch you.
That's right.
Even the mice, you'd probably have to do it because they want to squirm away.
You'd have to anesthetize me too, dude. I'm not putting up with this.
Well, in the SARS-CoV-2 human challenge, they didn't anesthetize those people.
Oof.
That's true.
They had them put their head back on the chair.
Oh, I remember. Oh, God.
Pipetted it in. Then they put a clothespin on their nose for 10 minutes so that it would stay there and not drip out, right?
They're even taking samples for diagnosis. They probably went through my cribiform plate several times.
Into your brain. That's what Daniel calls a brain biopsy. Okay, so you infect the ferrets. The chicken-derived viruses, the ferrets show few clinical signs. And then the human-derived viruses, they get severe signs, wheezing, dyspnea, coughing. And you know what dyspnea is?
Absolutely.
What is it, Dixon?
It's shortness of breath.
What's apnea?
It's when you...
Really?
I think it's when you stop breathing.
Yeah.
What's runny
when
nose?
You stop breathing,
that's
right.
Sleep apnea. What's runny nose?
Rhinitis?
Rhinorrhea.
Rhinorrhea.
Is it? What's coryza? Runny
eyes?
Eyes?
Yeah.
Coryza is nose.
Nose?
Inflammation of the mucous membrane in the nose. And there's another one for runny nose, runny. I don't know why I'm doing this, folks. Rhinorrhea is also a runny nose.
Rhinorrhea.
I thought coryza was a sausage from Latin America.
Coryza is
chorizo. Oh, okay.
Oh my gosh! So that's what the ferrets get, and then they look for virus, so they have to euthanize the ferret. And the human isolates produce a lot of virus in the nasal turbinate, trachea, the lung, and the brain. But the chicken H3N8 virus is only in the turbinate and the trachea, and not in the brain. And if you use a human H3N2 virus in a ferret, you only see it in the nasal turbinate and the trachea. So,
Vincent, can we stop here just for a moment and clarify something?
Of course.
You're using terms like ferret virus, mouse virus, human virus. This is the same strain of virus in various cell
there are two
lines.
kinds. There's chicken isolates of H3N8.
Yeah, right.
That have only been in chickens. And then there are isolates from people of H3N8.
So I'm asking if they've been sequenced.
Yeah, we're going to get to that.
We're getting there.
We're going to get to it. It's a good question.
Spoiler alert.
You must be a scientist.
I must be, but sometimes it's hard to tell.
So the human isolates, they do gross pathology, gross histopathological changes in the lungs, more viral antigen positive cells than in the chicken isolates. So basically all this together, the human-derived H3N8, they replicate well in the respiratory tract. They can spread to the brain. In mice and ferrets, they cause severe pathology, and that's very different from the chicken derived H3N8. So they're really different.
Yeah.
Okay. And this is
different.
telling us that, okay, they're different. They're not just the chicken virus in people. Somehow changes have probably happened in this H3N8 when it went through people.
Or they happened and that allowed them to go through people.
That's true. It could be that as well.
In gain of function?
Stuff happens. Not gain of function. It's just that viruses undergo mutation constantly, and it could be that in the chicken, a virus arose that could infect people, right? Just randomly. And then when it encountered a person, boom. It's all about
It's interesting that these are 3 separate occasions. And we need to re-ask this when we get to the genetics, whether I forget whether they're genetically identical in the different isolates. They're similar.
Similar. I don't think they're identical. Yeah. All right. Can they transmit? So the ferret is a good model for transmission.
I love these transmission experiments. I've always liked these for some reason.
So you can intranasally infect a ferret. Then you could put it in the same cage with another ferret, and that would be, that would be contact, right?
Mm-hmm.
They call that DC, direct contact. And then if you put the ferret in another cage, which is not too far away, so the air can blow the virus to it, that would be respiratory droplet transmission, a wireframe cage adjacent to another one.
I presume far enough apart so the ferrets can't touch noses or something.
Yes, absolutely.
Right.
I don't know if you remember this from ASV, Vincent, hearing about the ferret playpen model.
Yeah, I remember that. Yes.
There's a nice reading. There's a nice thing that Katie Boo wrote about that study, and it's a really funny discussion of that study too. So in that case, the ferrets can touch each other. So they're completely different models.
They put toys in, and ferrets play with the toys.
That's cool.
I think it's so funny when animals play with toys.
Well, especially because there's video.
Yeah,
you can watch this, and then they actually watch each ferret and look at infection in the ferrets, and did they play with the same toys and things like that.
All right, so the human H3N, well, first of all, the human H3N2, which is a real bona fide human virus, it's originally from the '68 pandemic. It's transmitted to all ferrets via droplets and via direct contact. Now, the human H3N8s, you can find them in the nasal washes from the ferrets, and only one of them spread by respiratory droplets.
Okay, yeah, I forgot. So they are different.
So the two human H3N8, only one does respiratory droplets. They both do direct contact, and they looked at antibodies in the ferrets to confirm that they actually got virus transmitted to them. All right. And they repeated that experiment. So basically one of these human isolates, HN4-10, can do respiratory droplets, but not the other one, interestingly.
And the chicken viruses can't even do direct contact.
They cannot do direct contact. What are they doing in terms of shedding? Let's see here. The chicken viruses, H3N2 viruses.
Well, they've got these, they do sample virus titer. That's, how are they sampling them?
Nasal wash.
Nasal wash? So I don't know if you call that shedding or not.
No,
probably.
So the chicken virus was only detected in nasal wash between days 2 and 4. So apparently that's not long enough to transmit in any way. Okay. So they can, one of the two can transmit from ferret to ferret, which is interesting. So now the question is, what changes are there in the genome? So they do genome sequencing. And we know that the hemagglutinin is important, and there are changes in the hemagglutinin that make it bind avian receptors or human receptors. And so one of these human isolates, CS1000, that was able to do the respiratory droplet transmission. Is that the one that does the respiratory? No, it was HK. The CS100 did not do respiratory droplet, and they have the avian signature of the HA. So it hasn't become able to bind human receptors, which are sialic acids of different types. Okay.
The PB2E627K was necessary for airborne.
Yeah, so that's different. So now they look. Let me finish with the HA. So the other isolate, HN4, that did go by respiratory droplet, that has human HA. The HA has. Got to put that down. The ability to bind.
Do it now.
Human receptors, alpha 2,6 human receptors. So a single amino acid change has occurred in that virus that is consistent with respiratory droplet transmission. Is that clear to everyone? You got that?
Mm-hmm. Yeah.
So one of the proteins that's a part of the RNA polymerase of the influenza virus, PB2, is known to have a change really important for going from avian to human cells. And remember, Brianne and Rich, you were both at the ASV in Madison, where we had, I know we had Andy Mealy.
Yeah.
And he talked about that mutation and amino acid change, which allows the polymerase to interact with a cellular protein. So it's really important for replicating well in human cells. E627K. It's another recognized adaptive mutation. These human H3N8, they have E627V. So CS has V and HN has K. And HN is the one that does droplets. So right away you see, it's got the K, which is probably...
That's lysine, right?
Yeah, it's amazing why it's doing it. So they look at the ability of these changes to facilitate RNA synthesis by using what they call mini genome replication assays at two different temperatures. So 33 and then 37, the temperature of the human. 33 is the upper tract, 37 is the lower tract.
Right.
And so they can show that K, the 627K, gives you really enhanced replication at 33 degrees, whereas the V, which is in the other virus, doesn't do it as well. And they introduced these. Sorry, Brian, go ahead.
I was going to say, they do that experiment in 293 T cells.
Yeah, right.
Do you think that they would see differences if they used chicken cells?
That's a good question, right?
Good question.
Theoretically, you would, right? Because that's a chicken
according to Andy.
Human adaptation thing. Yeah. They introduced these changes individually into viruses, and then they look at airborne transmission in the ferrets. And basically, the E, as Dixon said, E627K alone is enough to confer airborne transmission of the H3N8
So
virus.
as I was as we were creeping into these experiments in the paper, of course, I'm thinking the whole time about gain of function experiments, right? Because we're looking at what causes transmissibility in the ferrets. But this is an interesting spin on that whole conversation, because they start with a virus that they got out of humans that can already transmit in ferrets.
Yeah.
And they look at the genetics of it by what are in effect loss of function experiments. Okay? Because they take those viruses and swap it out, swap what ultimately is the culprit gene, swap it out for another one, and it's no longer transmissible. So if you want to complain about gain of function in this case, this is subtle, because this is loss of function.
Yeah, well, the other thing that's really important is that these are all changes in PB2. And usually when people are making arguments here, they're worried about making changes in HA and NA in, say, H5N1 that can lead to transmission. And here we're not in HA or in NA. We're in one of those other proteins that Rich mentioned at the beginning.
And importantly, these are all things that they pulled out of people. They're out there, okay? It's nothing that was created in the laboratory. They're just testing to see why it is, what the genetics are behind these properties that they're observing of these viruses that they pulled out of nature.
Look, the whole reason for the argument's validity is that nature is doing this already.
Exactly.
But we don't know where it is.
Exactly.
So if you do it in the lab and then go out and look for the same thing, you might find it.
Yeah.
What the hell's wrong with that? There's nothing sophisticated about that. That's brute force almost in looking. And they've actually defunded some programs now that were specifically, I just read about that recently in some place in Science, as a matter of fact, where they took the money away from people to go out and look. We don't want you to find that stuff. You find it, what are we going to do with it? Yeah, that's the attitude, right? You should probably make a vaccine against it.
This gives you some idea what changes you should be looking for in isolates that would be compatible with transmission. I mean, obviously there's some out there already that can transmit, but these only cause a few infections. So I think in the end you could probably need other things to change. There's not just enough by itself, right, to make a pandemic virus or even an epidemic virus, because the one isolate is got all the proper changes and it hasn't done anything. So clearly you need more.
That's the other getting ahead of ourselves here, but that's the other bottom line from this paper as well, is to reinforce that ferrets aren't people, because one of these guys transmitted seemingly pretty readily in the ferrets.
That's right.
By aerosol droplets, both of them by contact, and yet we had people infected with these, cared for by other people, and there was, as far as I know, no evidence of transmission. So ferrets aren't people.
And finally, they do genome sequencing to look elsewhere, and they say, can we find anything else that would, correlate with infecting in people? And they find one change in the HA, that is the one giving you enhanced binding to the human receptors. The E627K is there. They find a change in the NS1 protein. NS1 is an interferon antagonist, but they don't really see any differences in that property. They actually looked at interferon antagonism. So they conclude that, these viruses are heterogeneous. Oh, really? Yes, they are heterogeneous. That's what happens when RNA viruses reproduce. And there are only two changes, the receptor binding change in the polymerase, that seem to be enough to give you airborne transmission between ferrets. So they say the emergence of such a human adapted virus has the potential to spread in humans, but it hasn't. So, the last thing is very cool, because when Fouché and Kawaoka, actually there are two more things. Fouché and Kawaoka originally did their experiment where they took avian H5N1, which does not transmit by air among ferrets, and they passaged it in ferrets until they got isolates that could transmit. One of the phenotypes of they found amino acid changes in the HA protein. And they found that a key for transmission is for the HA protein to be stable at low pH. They said and I always thought that was a cool finding. And, people, the naysayers would say, we didn't learn anything from those experiments. We did. We learned that you need to be stable at low pH. So they look at the low pH stability of these H3N8 viruses, and they're not low pH stable. So maybe that's another reason why they're not transmitting among people. I thought that was a good experiment. Okay, so finally, H3N2 viruses are circulating, right? So H3N2, H3N8 is their cross-reactivity. So they vaccinate people with H3N2 virus vaccine, which we have. It's part of the flu vaccine. And they don't find any cross-reactivity between that antibody and the H3N8 virus. They do neutralization. They do hemagglutinin inhibition. And then they also collect sera from people of different ages, children, young adults, old adults, and find no cross-reactivity with these H3N8 viruses. So humans do not have any cross-reactive immunity against avian H3N8. So even though it's an H3, it's a different vintage. So it doesn't cross-react.
Vintage, good.
I got to tell you, when I was a student, I was talking with Jerry Schulman, who was an influenza guy. And I was asking him about some experiments. He said, well, what vintage of H3 did you use? And I thought that was just great. What vintage? So those are the experiments. We have H3N8 viruses in birds, including chickens. We get sporadic infections with humans. So the question is, should we worry? Why not? I think we ought to have a vaccine ready for H3 that's in H3N8. Why not? It's probably not that expensive to make it, right?
I'm totally on board with that.
The
only reason I would say no is that if we could make one that was more universal, that'd be cool. But
a universal would be cooler. Yeah, I agree. But they're doing H5 right now with mRNA and testing it just to be sure that it's going to be ready, right? Because
how many other influenzas are there out there that would meet these criteria that are circulating in chickens and occasionally show up in people? Do we do enough surveillance to know?
So H7,
probably a bunch.
H7N9, H10N8, H5N6, and H10N3 have all been known to infect people. And they say H5N6 can do contact transmission of ferrets, but not aerosol. Limited airborne in H7N9 and H10N3. So I think this is the most clear cut, this one H3N8, right, in ferrets in terms of transmission. So you're right, Rich. There's a lot of viruses out there. You can't make a vaccine against every one of them, right?
That's why the universal is so important. That would be
universal
good.
Would really be good.
Yeah.
And doing these kinds of experiments before you have the universal can help you to maybe triage. I don't know. It's a tough question. There's no answer, right? If you work on flu, you say, give me money to do it because you need to get money to do your experiments, right? But I don't know how many of these we need to have vaccines. Make a universal vaccine, folks, okay? Okay. I wanted to, let's see, do we have any short emails? Let's see.
Got a couple of really long ones.
All right,
we do.
Let's do couple of short ones. I wanted to do that first one, Dixon. Can you do Nicole's email?
Sure. Nicole writes, "Hello, my name is Nicole, and I'm a big fan of all the Microbe TV podcasts and have been for years. I am currently writing a thesis proposal on West Nile virus and remembered hearing that 90% of American crows died from West Nile virus. I couldn't remember where this stat was from, but then it hit me. TWiV! I looked back at the transcripts, and it was in the very first episode. I remember that. I'm amazed that this fact stuck with me for so long. And I was wondering if anyone over at TWiV has a source for this statistic so that I can cite it in my proposal." And then, Vincent, you picked out the same one that I was going to pick out because I was going to put that in there too.
Oh, good.
Or did Rich put it in there?
No, I didn't put it in.
Okay.
Yeah, so this was emerging infectious disease, West Nile virus, and high death rate in crows, American crows.
It says 2/3, not 90%. 2/3.
Yeah.
Still a lot of crows.
Listen, in some areas, all of them died. Right? It was 100% when they looked around New York City, for instance. But it varied. Of course, it did. By the way, I meant to say something when you were talking about ferrets transmitting the virus by nasal exchange. Crows do the same thing. And they found that out the same way. They infected one crow, put it into a cage filled with other crows, and the next thing you know, they were all dead.
Mm.
And then Cornell has the best website for this. So if you want some really good information that's up to date, Nicole, I suggest you go to Cornell University, upstate New York, not the medical school, and look for the West Nile virus project. And I think you'll find it online. And they've got current statistics and all that stuff. But I don't think anything is 100%. So 90%, if I said that, I was mistaken I was misled maybe by the fact that a lot of crows did die in New York City. But I don't think it was 100% or even 90.
All right, I put 4 short ones in here. All right, Brianne, could you take the next one?
Sure. Florian writes, "Dear Vincent, having just finished my PhD, I would like to take that as an opportunity to thank you and every member of the TWiX podcast universe. A friend suggested your online lecture on virology in spring 2020. I was shocked how little I knew about virology and what a cool field it is. Soon after, I became a regular listener of TWiV, TWiVO, and TWIM, the latter being close to my field of natural products from microorganisms. Later, I discovered TWIP, which made me watch the lecture series of Dixon and Daniel on parasites. Even though I cannot keep up with all your content, I appreciate all of them, each host and co-host, and of course your interview guests. It is so important to communicate science in days of people losing trust to standard care like vaccines. You teach the public how science proceeds, how research data are critically reviewed, how even established scientists never stop to learn and continue to be open-minded. Listening to your program expanded my knowledge and inspired me to approach my scientific discipline from different perspectives. For sure, you stuffed a lot of knowledge into my head. However, you also ended up on my head, decorating my PhD hat with a screenshot of TWiV, showing Vincent, Dixon, Kathy, Rich, Alan, and Brianne. I could send you a picture of that. Thank you for these 3 years of company, entertainment, and good science. Greetings from Germany, Florian.
Florian, send it.
Yeah, that ought to be
that would be cool.
An episode.
We'll all wear the same hat. Come on, we can all get a hat like that.
Send it. Love it. And hopefully you'll keep listening, right, Florian?
Absolutely.
And thanks for your compliments, Florian.
Nice. Thank you very much. Rich, can you take the next one?
James writes, "Hello. Thanks for all you do and learn much from weekly updates. Seems that post-infectious disease sequelae didn't get a lot of attention until the COVID pandemic. For a long time, these were dismissed, but have gained some attention in the past few decades. PTLDS, PVFS, CFS, and ME appear to have a lot in common with PASC. Thought you might have some comments on this. From all that I have read from the research, Occam's razor is not applicable here." And he links to a Stat News article that's titled "Long COVID is a New Name for an Old Syndrome." And I have not read this, but I would say based on the his description that things like ME/CFS and other hard to pin down lingering or chronic ailments might fall into the same pattern, and it sort of reinforces the notion that it might be worthwhile looking at an infectious instigator of those syndromes. But I haven't read this yet, but my guess is that's where it's going. Excellent. Thank you.
I think it's not the same, right? The article says, it's what is the title?
Long COVID is a new name for an old syndrome.
I don't think it's the same thing. I think every one of these post-viral syndromes has slightly different properties, right?
Yeah.
But we have post-acute viral syndromes before. Yes. I totally agree with that.
That's what we were saying after doing that paper. This is an appropriate letter for this episode. Is that, and as he rightly points out, we haven't paid a whole lot of attention to this until COVID, but it's probably not an uncommon phenomenon.
So I see in this article, there's a link to a paper in Science Translational Medicine. Core mitochondrial genes are downregulated during SARS-CoV-2 infection of rodents and human hosts. They're saying it's a possible cause of I have to look at that. Maybe this is a future TWiV paper. Have you looked at that, Brianne?
No. I have not.
Anyone?
No.
I'm going to look at it. Could be interesting, right?
Could be.
All right. Finally, Karen writes, good day. Brilliant and enlightening discussion on polio. Thanks for bringing in two more experts. Fleshed out the issues surrounding polio vaccination, infrastructure / climate cost, as well as issues concerning funding for continued research in irradiation / containment of the polio virus. Very enlightening for this layperson. Thank you, Karen. You're welcome. I'm glad you like it. Kostya Chumakov and Raul Andino talking about the new novel oral polio vaccines. That was fun. All right. What about some picks of the week? Hey, Dixon, what do you have for us?
Well, Vincent, I've got two very closely related subject matter, but in a very different light. The United Nations just recently concluded their climate change week, as if you should spend, maybe a week. That's enough. Of course, you're living climate change every day. And, I'm tired. I must say that I'm tired of large organizations coming up with these wonderful plans, and then no one buys into them. Nobody. And when I mean buy into them, I mean money and time and effort. And it's frustrating to see how many of these you can have and still not get anywhere. And so, as a matter of fact, I heard today that The Guardian, one of the reporters was on the Brian Lair show on NPR, and he said, do you know how much money the oil industry totally throughout the world is spending now looking for new oil? And the answer was $100 million per day to try to find more oil so they can do even more damage. And that leads me to my next article. This is the state of California's Attorney General, whose job it is to oversee, I guess, the policies that the state authorities, namely the governor all the way down through the health officials, have set forth, and he's the enforcer. So what he's done, he was on National Public Television last night, and he said the state of California is now suing all of the oil companies that have home offices in California, and a lot do, for lying to the public and telling them that there is no effect whatsoever of burning fossil fuels with regards to climate change. And they attended, he said, these companies made a point of attending all of these other large meetings like the UN and et cetera, to get all the facts down so they could couch their rebuttals in a light that made it sound as though it was the truth, but it turned out to be the same lie that the cigarette companies were telling us and lots of other industrial harmful activities were trying to pass it off. It's called greenwashing, obviously. This is how to change the narrative, in my view. I mean, start suing. So some students in Montana actually sued the state government for allowing fossil fuel companies to pollute the atmosphere and threaten their health. And and they got the lawsuit approved, and they're now going to go to court with it. And they can present their evidence. And there is a lot of evidence in favor of their point of view. And the same is true for California. The same would be true for Texas, Rich. The same would be true for New Jersey. The same would be, every state has a right, citizen right, to expect the best possible health for their citizens. And if an industry is threatening that, then you have a right to sue. And so I think
what's going to be the outcome of this suit?
That's a very good question. And the state of California's attorney general did not know, because no one has ever done this before.
But
they don't have any other.
From a practical point of view, the analogy with the tobacco lawsuits is appropriate. Okay, the outcome is that if you're successful with a lawsuit, you get money, and you use that money to help try and remediate the problem. You also put the perps on notice that they got to get their act together.
The thing is, when you look at the opioid industry, right, and the amount of money that they've forked over as a result of their lawsuits, to even have that much money tells you that that was the tip of their iceberg. They're rich beyond their imagination. And the oil companies are in the same position, and the cigarette companies didn't stop making cigarettes. They're just not selling them as much as they used to. But it's a different world, and I don't know what else to do. Basically, right? You can write a book or whatever you want.
It's one of several things you can do.
Yeah. So all my picks from now on are going to relate to the environment. That's what I'm going to do.
All right. Okay. Thank you, Dixon.
You're welcome.
Brianne, what do you have for us?
I have something that I read earlier this week, and I immediately clicked to see if there were show notes because I knew I was going to pick it. It is for the cats of TWiV, including one who may have visited us earlier. So this is called COVID Drugs Are a Miracle Cure for Cats in the Atlantic. This talks about the FIP coronavirus that impacts cats. In particular, a lot of free-roaming cats in Cyprus who are currently having a pretty massive FIP outbreak. And it turns out that it was found that some of the drugs that have been used for or developed for SARS-CoV-2 also work for FIP in cats. In particular, we remember molnupiravir, which sort of has fallen out of favor. While Paxlovid has been the one people are using instead, there are large stockpiles of molnupiravir that have been held in a number of places, and those large stockpiles have been sent to Cyprus and are actually stopping the FIP outbreak in cats.
That's very cool. I like that. How do they give it to the cats?
Wow.
That is not really clear.
Okay.
They force feed them out.
But they seem to be having a huge change in the number of FIP deaths in the cats.
That's very cool.
And so I really liked the idea of unintended consequences with all of this virology that's been going on in the past couple of years.
So I talked to a couple of people about this lately. I was up at Cornell last week at the vet school, and there was a lady there who knows all about this and said that they're trying to convince Gilead, who makes remdesivir, to allow it to be used veterinary use. And they're being resistant for some reason, but she said she's on a crusade.
Yeah, it mentions remdesivir in the article.
And then on the stream the other night, we had a person who works for a cat shelter in New Mexico, and she said they give it to them intramuscularly, the remdesivir, because otherwise it hurts them if you do IV for some reason, right? So... Yeah, she said it's a miracle. It's amazing. It works.
Wow.
That's very cool.
This cat that they have pictured in this article looks like a pretty tough character.
It does, yeah.
It's cool. Rich, what do you have for us?
My pick is a book called American Prometheus by Kai Bird and Martin Sherwin. This is the biography of Oppenheimer that the movie was based on. Okay. And in fact, it parallels the movie. The movie obviously really took off on this book. I would put this book right up there with The Making of the Atomic Bomb as one of my all-time favorites. It's riveting. It's really, and I don't know why, because some biographies I read and they're snoozers. But this one, he's an interesting person. It's an interesting topic, but I think a lot of it has to do with the writing. Okay. It's an easy read. And it's really compelling. It's very good. In many ways, it's a tragedy. It's quite something. But if you've seen the movie, I saw the movie first. There's always a question in this case: should you see the movie first or read the book first? I have to admit, reading the book, every time there's talk of Leslie Groves, I'm picturing what's his name, Matt Damon. So, but at any rate, the movie's great too. Have you guys seen the movie?
No, I haven't seen it. No.
I haven't. I really want to.
It's really good. I thought about making a double pick, but what the heck? This does that.
I'm reading Making of the Atomic Bomb, and the parts on Oppenheimer are just riveting. I mean, the guy, as a kid, he was amazing, right?
Yeah.
And then, he was kind of a sickly kid growing up in Manhattan. His father sent him out west to work on some thing, and he became tough,
Yeah, shades of Teddy Roosevelt, right?
Oh man,
shades of that cat from the other picture.
What an interesting fellow. I mean, a lot of these physicists.
If you make it through Making the Atomic Bomb and you're still and you're hungry for more, this book is for you. This is really good.
I mean, these physicists were all such interesting characters.
Well, it's such an interesting time. It's amazing because, there was a large percentage of very plugged-in physicists who were saying that fission couldn't be done.
Right.
And then somebody did it, and immediately what came to their mind
Enrico
is,
Fermi did it.
Oh my God,
Fermi
bomb. Okay. They recognized immediately the potential of this for a release of a lot of energy.
Yeah.
There was
it took an Italian to show them, Vincent.
Yeah.
Fermi. Yeah. Well, Leo Szilard said he was crossing a street in England and he had the idea, but he never did experiments, right? He would just think about it. But so I think it's really interesting how Einstein became like a time cover person, the most famous scientist in the world, right? And when Marie Curie died, he said she's the only person who got famous and didn't let it corrupt her. The only scientist, It's very cool. I mean, all these quips, it's just great. I love it.
Great stuff.
My pick is part of Carl Zimmer's website where he posts the articles he writes in the Times, National Geographic. I didn't realize this, but we did his paper on his article on TWiV yesterday, which is about, there's some controversies in some findings of bacteria and fungi in cancers, right? And he wrote an article summarizing that. And I said, unfortunately, it's behind a paywall. And Michael Schmidt said, no, he posts them on his website. So I'm going to put a link here to the page. It's called Articles. And he posts stuff from the Times and the Nat Geo and so forth. So it's the same article and it's free. So probably not all of them, but here the researchers dispute high-profile discoveries of cancer microbes is there. So if you're looking for a Carl Zimmer article, you should check there. It's cool. I didn't know that he did that. Cool. Thank you, Carl. Anne has a listener pick, which is, she wrote to a scientist about her fatigue. It inspired a breakthrough. This is the Washington Post article. And kind of the backstory leading to the mitochondrial finding that I just told you about. So that's cool. And it was kind of inspired by somebody who wrote in and said, I have this problem. So that's cool. Thank you, Anne. And Alan writes, a parasitic wasp that can drill through a petri dish to lay an egg. This may represent speciation in real time with lovely videos of the action of the ovipositor drill used by a small subset of Euplemus Messeni wasps enjoy.
Is this a glass or a plastic petri dish?
Plastic.
Plastic.
Wow.
Okay.
And so that's a Times article by Darren Incorvaya, but you can see the video. You can see that with them drilling through the plastic.
My God.
Wow.
Cool.
So you might want to ask, why did the Euplemus Messeni wasp drill to the other side?
Wants to lay its eggs.
To get to the larva that he's going to put the egg in, or she's going to put the egg in.
It's a lady. She's laying eggs.
It's a woman. It's a lady. It's a lady bug. That's right.
All right.
That's the ovipositor. That's the egg-positor.
Yeah, ovipositor. It's a good name. We use specific words because we want you to know what we're talking about.
Indeed.
Right?
Indeed.
We use capsid instead of box. By the way, capsid is Latin for box, but we say capsid. Everybody knows what it means on this program anyway. But if I said box, it could be anything.
Exactly.
So that's why we use hard words, not to confuse you. All right.
That's also why we argue about them.
Yes.
Like, how do you define infection?
Right.
It's a good question.
What is life?
That's a good question. All right. That's a Schrodinger book. What is life? Do you know that?
Speaking of cats.
Yeah. It's a cool part of the making of the atomic bomb where, Schrodinger had published his uncertainty principle. And then he went to a meeting. I think it was Niels Bohr who just kept at him and saying, it's not right. It's not right. And he finally backed down. But he is right. Because when you study things, you perturb the systems, right?
It's not a belief system. Science is not a belief system.
I like these meetings where all these people go and they talk and they argue. We don't do that. We just give a talk and go home,
Well,
at meetings.
Sometimes we argue a little bit.
Maybe afterwards in the bar, but at the actual talk, nobody.
It's how strong the evidence is. How provable is that evidence, right? When you have to explain it to somebody who doesn't know anything.
Somebody gets up and gives a talk, and then afterwards people say, he's just a fool.
Exactly. Well, the classic argument, the classic moment on one of these was when Stephen Hawking was giving a lecture at Cambridge on his theory about black holes.
Mm-hmm.
And he was about halfway through, and somebody in the back of the room, an astrophysicist, said, "This is all bullshit," and he gets up and he walks out. When Stephen Hawking turns to the audience and says, "Was it something I said?" You're damn right it was.
It's funny. All right, TWiV 1047. Show notes are at microbe.tv/TWiV. You can send your questions and comments to TWiV at microbe.tv. And again, if you like what we do, if you enjoy these shows, please support us. We really do need your help. Microbe.tv/contribute. Dixon Despommier is at trichinella.org and thelivingriver.org. Thank you, Dixon.
You're welcome, Vincent, and everybody else. Thank you for being who you are. I appreciate being a part of the group.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time. Next time, I'm bringing my giraffe. So you guys, I got to show you Alan because he's my buddy. Right. And I think this probably ought to be tagged as one of the all-time silliest TWiV episodes.
Well, why not? I'll bring some of my grandson's animals that we've bought for him also. You'll like them. My wife does a wonderful imitation of each one of them when she plays with him. So maybe I'll get her to bring them in and she can introduce them to the group.
Brianne Barker's at Drew University, BioProf Barker on X. Do you want to go by X?
Whatever. Thanks. I learned a lot.
You should go to the page. It says X, but I still like
true,
Twitter. I think Twitter is cool.
The app says X.
Uh-huh.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You're listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.